Hi, Greg. Welcome to the Big Unlock podcast. It's great to have you on the show today.
Thank you for having me, Rohit.
Absolutely, Greg. So, we'll begin with some very quick introductions. I'm Rohit Mahajan. I'm the CEO of Demo Consulting and Big Rio. And Big Rio is a Boston based consulting company, and Demo does strategy consulting for healthcare providers and payers as well. And Big Rio works with a bunch of pharma companies on AI and data and analytics initiatives as well. So I think we are in for a treat with the listeners. Over to you for your introductions, Greg.
Thanks, Rohit, and thank you for having me. Yeah, so thank you for inviting me. I mean, I understand that most of the time your focus is on healthcare, um, and biopharma is sort of healthcare adjacent, and I understand that it has some crossover interests. So I appreciate the invitation to talk to your audience. So my name is Greg Goldmacher. My current position is that I am Associate VP of Clinical Research at Merck Research Laboratories. I'm head of the clinical imaging and pathology function.
Okay.
So my team oversees the use of scans and tissue in assessing trial outcomes, in clinical trial endpoints.
Right. And as we all know, Greg, there are so many, I understand that there might be a million clinical trials at this point in time. So I think the number is a very large number, possibly definitely north of 500,000, maybe approaching a million clinical trials. So what kind of clinical trials do you focus on with your group?
So my group is has a pretty broad base, and we um, support trials across every therapeutic area.
Hmm.
Of course, so we go basically where the endpoints are. What is an endpoint? An endpoint is a
Hmm.
measurement. It's a measurement of whether a clinical trial has met its objective. So,
Hmm.
you make a measurement in one group of trial participants, you compare it with another group of trial participants, and then you basically you see whether you have shown safety and efficacy. Um,
Hmm.
the majority of trials where clinical imaging is used for endpoints, that's in oncology.
Hmm.
Because, of course, kind of just intuitively, you can think about, what do you measure to see
Hmm.
whether a cancer drug is working? Well, are the tumors growing or shrinking over time?
Yeah,
And the way you do that is by doing a series of scans.
right.
So that's where so there are several hundred clinical trials that my team supports.
I see.
But and with a large majority of them are in oncology, but we also support central nervous system, immunology, cardiorespiratory, metabolic disease, a variety of other therapeutic
That
areas
is
as
that is
well.
fantastic, Greg. And what I do understand, from whatever I know, my limited knowledge on clinical trials, I do know that it's a fairly long and an expensive process because human beings, patients are being involved in clinical trials, so it's usually a big hurdle for the biopharma companies to complete clinical trials in such a manner that the drug can go to market, right? So could you tell us a little bit more, for listeners who may not be aware of this long and expensive process on how this works, like very high level? Yeah.
Sure, sure. And I'll explain. By the way, I have to preface everything that I'm saying here, but that I'm speaking on behalf of myself. I'm not speaking on behalf of Merck, and I'm not going to discuss any specific Merck products or programs or specific Merck trials. So
Great.
I'm going to speak in broad ways about biopharma, about the processes, and then we can get into the use of technology and AI in this.
Absolutely. Yes.
So I guess let's start with the big picture. There's the preclinical process where molecules are designed, are evaluated for the molecular properties, and are evaluated first in vitro and then in preclinical in vivo models, so animals. A lot of it is looking for things like pharmacodynamics, pharmacokinetics, the distribution of the drug in the organism, whether it's getting to its target, um, and you do preclinical safety, those sorts of things, and get some initial idea of dose. Then comes the clinical part. So once you move into clinical trials, phase one trials are largely focused on safety and establishing dose.
Hmm.
then there's transition into phase 2, where you're looking at nailing down the final dose, and gaining ideally some signal of efficacy,
Hmm.
and safety. Safety information is collected throughout. Then in phase 3 trials, that's where you really are nailing down your efficacy. Those are often referred to as pivotal trials. And although phase 2s may be pivotal as well, so data from phase 2s, if they're appropriately designed, and phase 3s can be submitted for regulatory approval, to authorize for marketing of the drug. And then
Yeah.
occasionally you could also even have phase 4 trials, which typically happen post-approval, and they collect additional safety information or information on the efficacy of drugs in the sort of real world setting.
Right, right. So that's a great high level intro, Greg. Thank you for that. So before we segue into, and it's not too long in any podcast before AI comes up, right? So we are
Of
going to
course.
talk about the use of AI and all the new technologies in this process of, the clinical trials, so that it can be made more efficient and cost effective, and faster perhaps. So what are some of your thoughts on that? And before that, if you could just get some insights on into how did you get to this position where you are at, and how did you start your role? And I understand that you trained as a physician as well, so would love to hear that story before we get into dive into AI side of things.
Okay, sure, Rohit, thanks. So I started off thinking I was going to be a basic science researcher and work in a lab. And then in college, at some point, I was working in a lab and doing a summer research position, and somebody said, you should get an MD PhD rather than a straight PhD because you'll have an easier time getting research grants that way.
Awesome.
So I went ahead and did that. I did an MD PhD at UT Southwestern. My PhD was in neuroscience, and I briefly thought about being a clinical neurologist for my clinical training, and then thought, I'd be too frustrated because this was in the mid 90s. The bread and butter of neurology was stroke and neurodegenerative disorders. And in both cases in the 90s, you basically, you make the diagnosis and then you say, good luck, go see occupational therapy, because there was really nothing disease modifying. So
Mhm.
instead, I went into diagnostic radiology. I came back to the Boston area, did my residency in diagnostic radiology, and then I was a fellow over at Mass General in neuroimaging, was doing a bunch of stroke imaging research and got involved in a clinical trial. So I was helping to run an NIH-sponsored clinical trial, but my mentor got me involved in a small industry-sponsored trial where they were using it was a thrombolytic drug, and they were looking at reopening of the vessel of the artery on CT angiography to look at to get an initial sense of efficacy. And I
Mhm.
effectively became the core lab as a fellow for this trial, which a couple of years later, long story short, I was doing more stroke research at Jefferson in Philly, and realized that the lab is, I can do the lab, but it's not really my jam.
Yeah.
And at that point, it turned the chief medical officer from Icon came and gave a talk at Jefferson about things you can do as a physician and scientist in industry, and mentioned that they had a radiology core lab. And this is an ecosystem that I had no idea about. You never hear about these things in your medical training.
Mhm.
So I actually went to Icon for 5 years, was actually effectively retrained as a cancer imager, because that's where a lot of the endpoints are, as I described, based on imaging. And then in 2015 came over to Merck.
Mhm.
So supported a portion of the cancer imaging portfolio for a while. And then as organizational changes happened, took on supporting all clinical imaging across the entire portfolio, all therapeutic areas. And then later we added clinical pathology as well, because the assessing outcomes using pathology in a lot of ways conceptually functions the same way,
Yeah.
but it's relatively newer to have registration approvable endpoints based on pathology. So, in imaging, we've worked out all the operational aspects of it over a long time. So we've kind of
Yeah.
added in, we're kind of transposing those processes into that space.
That's true.
Um,
That's true. Yeah.
Now going over to the technology side of things, so I like to say in radiology, we were doing AI research before ChatGPT made it cool.
Of course. Yeah.
Um, because of course, what is AI? AI is pattern recognition.
Yeah.
And visual pattern recognition, it's something that we as humans do all the time, right? You show us a picture of a dog or a cat, you
Yeah.
can say whether it's a dog or a cat, you're very confidently, without
Mm.
being able to describe explicit rules for what measurements, will tell you whether it's a dog or a cat, right? So in radiology, there's similar sorts of things where models can see patterns. Now, the cool thing is that models can see patterns that human eyes can't see, no matter
Yeah.
how good a radiologist you are. And so there's a lot of interest in that. And so, sometime after, shortly after I joined Merck, I started pulling together people, other enthusiasts
Yeah.
from data science, from statistics, clinicians, biomarker folks, regulatory digital folks, I mean, all sorts of people from across the organization who have come together to do research in this space.
Great. Great. So, what are some of the things you feel, Greg, given your deep experience on the clinical trial side of things and the AI as well, and now with the new chat GPT, what are some of the things that you are seeing which are applicable in the clinical trials world?
So there's kind of a couple of broad categories. Um, let's start with one that is sort of the easiest and most obvious business case for
Mm-hmm.
the use of AI in the clinical trials world, and then we can segue into things that are
Yeah.
more maybe a little bit less obvious, but I think scientifically and from a business strategy point of view, very interesting.
Mm-hmm.
So the easiest place to make the business case is, clinical trials are expensive, and a lot of it is human resource cost, people. So there are a lot of applications of AI which everybody can think of because we're starting, everybody's starting to write things using AI, et cetera, AI tools. Is essentially efficiency. So now in the preclinical space, AI is being used for things like genome searches for target identification,
Mm-hmm.
for drug design, assessing protein folding and things of that nature.
Mm-hmm.
But now, once you get into the clinical space, there's a lot of use of AI to support clinical operations. That includes things like creating documents, protocols, clinical study reports,
Mm-hmm.
informed consent forms, reports of various kinds.
Mm-hmm.
There's also AI analysis of various reports. A huge amount of data gets collected, and manual review of all that data is extremely labor intensive.
Right.
And that is an area where AI can absolutely help. Just as one example of a kind of effort that's often done manually is there's a million kinds of reconciliation that you have to do in clinical trials where data is gathered in one place and there's also data in another place, and you have to make sure that things match. Either data has been transmitted or processed, and you have to do this reconciliation. That can be an extraordinarily labor intensive process manually. And that is one where both, AI, for kind of just general AI use, and specifically agents, where you can say to an agent, retrieve data from this place, compare it to this, do the comparison, take an action based on the comparison, such as
Mm-hmm.
issuing a query to a site to ask for clarification of some data that's been recorded, or where
Awesome.
there's question
Yeah.
about the data. That kind of stuff is a great use in terms of efficiency.
Mm.
And that is the most kind of the most straightforward business case. I think everybody sees that, and that's being adopted, not obviously not just in biopharma. This kind of use of AI is used across many industries, but in clinical trials in particular, where reports and documents and reconciliation and analysis are so important, that would be a valuable tool.
That's amazing. I think that's great to know. And then, oh, so in drug development decisions, if we talk about AI applications in drug development decisions and automating tasks and making strategic decisions, for the organization. What do you think about that aspect, Greg?
So that's one where, so efficiency, as I said, that's kind of the obvious use case. the slightly less obvious, but I think at least as impactful use case. And of course, obviously, I'm speaking, from my
Mhm.
parochial perspective as somebody who focuses on imaging assessments.
Yes.
But thinking about endpoints, right? Endpoints are measurements. Why do you make measurements? To make decisions.
Mm-hmm.
AI allows us to make better measurements for better decisions. Let me explain what I mean.
That's fantastic. Yeah.
So, as an example, let's use cancer. All
Mm-hmm.
right. So, the traditional way of assessing whether a cancer drug is working
Mm-hmm.
in a clinical trial is that you do a scan, let's say a CT scan, before treatment starts.
Mm-hmm.
You find all the tumors, you pick out a few of them that you're going to measure. Then
Mm-hmm.
you kind of add up their measurements, and then you do multiple scans during treatment, and you look at those tumors. And if they shrink, that's called a response. That's good. If they grow, that's called progression. That's bad. And so what you do is at each assessment time point, you basically look at all the tumors, apply some very simple math and logic rules to classify them as, a complete response, partial response, stable disease, or progressive disease. That's kind of treatment failure.
Mm-hmm.
And then you take a look at all those responses, and you extract an endpoint like objective response rate or progression-free survival in using sets of rules like the response evaluation criteria in solid tumors, RECIST. That's like a very standard tool in clinical trials. Okay, so what I've just described is the traditional method of assessment. Now, what we really care about is not tumors growing or shrinking, right? What do we actually care about?
The patient
Survival.
better. Yeah. Yeah.
Does the patient survive, live
Yeah.
longer, right?
Yeah. Yeah.
So these kind of traditional assessments, like objective response rate per whatever set of criteria, like RESIST, that is a good predictor of survival
Mm-hmm.
at large N.
Mm-hmm.
That is, when you have a large a population of subjects,
Mm-hmm.
improvements in objective response rate really indicate improvements in survival. The problem is that as the N gets smaller, that
Mm-hmm.
correlation gets worse.
I see.
And
Okay.
there are lots of examples. Anybody who's done cancer drug development for any length of time can think of examples of
Mm-hmm.
drugs that looked good based on response rate in those early phase trials in phase 1B, 2A, where you're starting to look at efficacy, but you've only got dozens of subjects' data, not
Mm-hmm.
hundreds.
Mm-hmm.
And they looked good at that time, and then they went to phase 3 and failed to improve survival. And that's a terrible outcome for patients, obviously, because they don't, but it's also a huge waste of resources.
Yeah.
And a phase 3 trial, we're talking, $150 million,
Yeah.
right? So if a phase 3 trial fails, that is a big bad problem.
Yeah.
But the decisions have to be made. All those pipeline decisions are made early on in
Mm-hmm.
that phase 1B, early phase 2, where you're deciding on dose. And by the way, that's another application of these things, is picking the right dose of a drug. So the way that, kind of traditional dose finding is you crank up the dose until the patient keels over from adverse events, and then you back off a level, and you call that the maximal tolerated dose. There's a
Mm-hmm.
variety of reasons why that may not be optimal, even from an efficacy standpoint, let alone balancing kind of risk versus efficacy, safety versus efficacy trade-offs.
Yeah.
So if you could meaningfully compare survival,
Mm-hmm.
groups of 30, 40 patients, which is what you do when you do dose expansion cohorts in kind of early phase development, you could get to a confident dose that you move forward
Mm-hmm.
into late phase trials. So now here's where AI really can come into its own
Mm-hmm.
in a couple of different ways. And again, speaking from my perspective as somebody who looks at pictures, right? Scans contain lots of information that isn't immediately visible to the naked eye.
Yeah.
So models can see patterns, pixel patterns. When you look at the image of a tumor, there's a
Mm-hmm.
lot more information than just how big is the tumor.
Mm-hmm.
There are pixel patterns that you can extract with models that may correlate with necrosis or vascularity or inflammation, that basically reflect aspects of the tumor microenvironment that
Mm-hmm.
are not appreciable with the eye,
Mm-hmm.
but can be measured by a model. And that if you have a training set where you can associate these pixel patterns
Mm-hmm.
with some gold standard of truth, obtained, for example, through a biopsy, then you can do non-invasive assessment of the tumor microenvironment.
Right.
And that would allow you to use that, for example, as a pharmacodynamic biomarker. So, for instance, if in drugs that are sort of in the immuno-oncology space
Mhm.
work better when tumors are inflamed, when there is lymphocyte infiltration and inflammation in the tumors.
Mhm.
Well, if you could measure that non-invasively across the entire patient, meaning all their tumors, not just one tumor that you can hit with a needle, then
Mhm.
you could use that to assess whether a potential combination partner for a drug that works better in inflamed tumors is a good combination partner, right? If you give the drug and all the tumors light up with inflammation, hey, that's a potentially good drug partner, combination partner.
Right.
So that's kind of and there's also the potential for looking at these early changes in tumors and directly predicting survival rather than having to basically say, well, does it predict tumor shrinkage, and then shrinkage predicts survival, right? So that's one possibility. Another
Mhm.
is there's a way of looking at tumor at the it's just, so a downside of systems like resist is that they're essentially saying, look, if there's a fixed percentage change in the size of the tumors, that's a response. Well, it doesn't take into account the kinetics of this. And there are ways of basically a common an approach that there's a lot of academic evidence for is you say you take the total tumor burden and you say, let's assume that it consists of a treatment sensitive fraction that exponentially decays during treatment and a treatment resistant fraction that exponentially grows. So if you take total tumor burden, plot it over time, and say, okay, computer, fit me a curve that assumes that it's the sum of a decay curve and a growth curve governed by a decay constant and a growth constant, one of which may be zero. But then you back out that growth constant. That growth constant has been shown, as I said, in a lot of academic work, to be much better associated with survival than traditional metrics. And that should intuitively make sense, Rohit, right? Because how do
Mhm.
tumors kill you? By
Mhm.
growing.
Yeah.
And
Yeah.
so characterizing the behavior of the treatment resistant fraction of the tumor,
Mhm.
it makes sense that it would be a good predictor of survival. Now, here's the challenge.
Mhm.
When the system of assessment that I talked about before, like RESIST, it depends on taking a few tumors and just measuring them in diameter and adding up those diameters. The problem is anytime you sample,
Mhm.
there is an assumption you're building in, and that is that the outcome of interest, survival, is driven by the average. That's when you sample a population. So when you pick out a few tumors out of all of a patient's metastases, you're assuming that the average is what drives the outcome. But that's not true. What
Mhm.
kills the patient isn't the average tumor, it's the worst tumor. It's
Yes.
the most treatment resistant, most aggressive, most rapidly growing, and its clones, its descendants.
Yeah.
That's what's going to kill the patient. So the problem is, if when you were looking at the initial scan, and there's no way to know which of them is the worst tumor.
Yeah.
If you didn't happen to pick it as one of the handful of ones that you're following quantitatively, you're blind to it.
I
So
see
you
what.
really want to measure everything, number
Right.
one. Number
Yeah.
two, you want to ideally, there's been academic work showing this, you should not, rather than just slapping a ruler and measuring a diameter, you want a full 3D outline of the entire tumor. The
Mhm.
problem is radiologist time is expensive and image analyst time is expensive. And
Yeah.
finding every tumor and drawing it in 3D
Yeah.
is not scalable. It's too expensive.
Right.
So, however, AI can do it,
Yeah.
right? There are now good AI tools there still aren't tools that automatically find every tumor. There's a lot of work being done in this space.
Yes. Yes.
But once you point to a tumor, it'll draw it in 3D. And then a machine can, co-register all the scans across time based on those initial identifications, draw in 3D,
Yeah.
propagate it throughout all the tumors, make all the measurements, and feed that into your modeling. Right?
Very, very
So
interesting. Yeah.
the idea here is with that combination of directly AI assessments and
Mhm.
assessments that are sort of assisted by AI or made scalable by AI,
Yeah.
you could get to a place where you get better measurements of efficacy which let you make better decisions about a drug pipeline, about whether to move forward with a combination, with an asset or a combination of assets,
Yeah,
or whether to, and what dose to use when you do that.
that's a great explanation. Thank you for that, Greg. So, as you mentioned, that a lot of this is exploratory and it's academic in nature. And when do you see it moving into industry grade regulatory practices? Because, pharma, biopharma, highly regulated industry segment. So what are some of your thoughts around that space and any FDA, insights that you can provide?
Sure. So what I would say is, first of all, with regards to the kinds of measurements that I've talked about, you
Mm,
notice that I talked about using them in early phase decision making. This is
mm,
purely internal decision making.
mhm,
Now, as you move forward, you and pharma decision makers are conservative by inclination. They're
mm,
risk averse. So
mhm,
you have to do a lot of validation first. You got to validate internally, and the initial uses are internal uses. Now, as evidence starts to accumulate, you can start building these kind of exploratory assessments, or you have these kind of more advanced assessments
mm,
into phase 2 trials as exploratory endpoints.
mhm,
And then, so in order for the FDA to accept a measurement of any kind, whether it's AI based or not, as an outcome in a trial, there needs to be a strong evidence that those measurements are in fact strongly associated with clinical benefit. So essentially that's the process. Is you start with using it for internal decision making, then
mm,
you build it in as exploratory endpoints, then you start accumulating that data, you submit that data to regulators as part of the overall package,
mm.
and as the experience with it builds up, you can
Yeah.
sort of start thinking about using it initially maybe to supplement regulatory decisions, like um, you know supporting a breakthrough designation, or supporting what in the US would be called an accelerated approval,
Mm,
which is or like a provisional approval, effectively. And then someday potentially getting to primary endpoints. Now, the FDA put out a guidance in January of this year on the use of AI in regulatory decision making.
mhm,
And they laid out a whole framework for building what they called it a credibility assessment framework,
mm,
where essentially you have to define the context of use very clearly. Then you have to assess the model risk. So if you're going to be making decisions about treatment, about endpoints, about whatever, based on the output of an AI model, you have to consider things like, is it the only thing that the decision is being made on the basis of, or is it just a contributing factor? And what's the potential risk of getting it wrong, right? So if you're making a decision about treatment on the basis of an AI output, that's
mhm,
a very, that's high risk, right? You get the treatment wrong, that's
mm,
terrible for patients. Whereas if the decision making is not as directly about care, the level of risk is lower. So you have to assess the model risk, and then you have to generate evidence that is sort of commensurate with the level of risk. And
sure.
the evidence itself has to be generated in intelligent ways. You have to watch out for pitfalls,
Mhm,
like various forms of sort of data commingling or circularity. That is, in various subtle ways, if you train a model on data and then test it on data that's overlapping, that can give you results that aren't accurate, that don't really generalize. So
mhm,
you have to think through all of the pitfalls,
mhm.
um, to build a base of evidence that the FDA can sort of accept as commensurate with the model risk. And
Right.
the bottom line is, right now everybody's in kind of exploratory mode and figuring out how to do this.
Mm,
The draft, the guidance that came out from FDA in January was a draft guidance,
I see.
but these guidances from FDA can remain in draft for years
Right.
and reflect kind of the latest and greatest thinking from their end. There's been responses, there's various published analyses, of course. Pharma, and bio have generated organized responses to this draft guidance, but that is where I would suggest people go to read about the FDA's thinking about this. But the bottom line is, whatever you're thinking of doing with AI, if you're in biopharma and you want to start with you want to bring it to regulators eventually, it's to engage early, to
Yeah.
engage with them early, to sit down and say, here's our plan, here's the technology, here's the data we've trained it on, here's how we've done validation, here's the
Mm-hmm.
context of use, here's the risk,
Mm-hmm.
and essentially use that to build that credibility assessment framework.
That's amazing. And then, Greg, touching upon what are some of the challenges? Like you're AI innovator in a very large biotech, biopharma organization. So, what are some of the challenges that you run into in such like environments, and how do you overcome them? What are some strategies that you have followed successfully? If you would like to share about that
Sure. So it's a great question. One thing that big pharma has in massive amounts is data.
data.
If you're in a pharma that has run a large clinical development program, you're drowning in data.
Mm-hmm.
That some of the challenges are around, some of them are around data standardization.
Mm-hmm.
So a lot of times you will have legacy systems, and a big pharma may have, dozens of legacy systems that collect data, and that can't necessarily talk to each other. And when you want a model to go in and look at data, it really helps to have a data standard. That's actually something that was really helpful for radiology, why we started doing AI research so early, because radiology has this data standard called DICOM. No matter
Yes.
whether you have a scanner from GE or Philips or Siemens or Toshiba or Canon or whatever,
Mm-hmm.
they all generate DICOM data. And by the way, the reason that that was possible is that there were only a few major equipment manufacturers, so their data engineers could get together and say, here's a data standard. Everybody else kind of has to stick to it. But a lot of other kinds of data aren't standardized that way. So that is an important challenge. And actually, that's a market opportunity for any companies who want to go in
Mm-hmm.
there and basically create tools. And AI would be actually a really useful thing for this, to go and interconvert data and harmonize it and bring it into one environment where it can be used for training and validation. Another aspect, though, is kind of the sensitivity around access to data and consent of patients who consented to a trial. So you really need to be aware of the fact that clinical trial data is sensitive data.
Mm-hmm.
And you have to think carefully through how to put in risk mitigation strategies to make sure that you are not going to analyze that data in some way with AI and generate an output that is in some way confusing or undermines essentially what is known about a medication and so forth. Now, the other important aspect, and again, there's various ways of doing this, but you have to think about that in advance, is
Mm-hmm.
how do you de-risk this
Mm-hmm.
kind of use of the data?
Mm-hmm.
Another important aspect, as I said, is consent. So did the subjects in a trial consent to that use of their data? And that's essentially the mitigation for that, is to work with the people writing the informed consent language to clarify that these kinds of additional uses of data beyond the use in that specific trial are permissible. And then finally, the other, so aside from data, there's the people side.
Yeah.
So, the challenge is that a lot of times data scientists and AI engineers are absolutely brilliant, but
Mm-hmm.
what they don't understand the problems that need to be solved. So they may come up with solutions, sort of solutions in search of problems, or solutions that are difficult to apply. So that any organization that builds a team that is doing work in AI needs to bring together the people who understand the challenges. These are the clinical
Yeah.
developers and the strategists, right? With the people who understand the tools, data scientists, statisticians. Statisticians are really critical because all of those pitfalls about overlapping data, overtraining, and things of that nature, statisticians are really key for that. So
Mm-hmm.
bringing together people with, so what you want in an AI team is a really multi-dimensional view of the problems and of the tools. And then you collaborate on developing the tools to solve the problems.
Yeah, that's a great insight, Greg. So as we come to the end of the podcast, Greg, uh, what are some of your thoughts for the future? Like when you look into the, you know, crystal glass, what do you see that might be some, you know, small or big or, medium-sized changes which might be coming our way?
So what I see is right now, so there's always, like, for any technology, there's the hype cycle,
Yeah,
and people are, very enthusiastic about it'll do everything. It'll
yeah,
walk your dog and
yeah,
run your clinical trial and butter your bread, whatever. It obviously, AI is a set of tools. It's a set of tools that is potentially very powerful, but you have to use it with great caution, because how you train and how you validate and all these things are important caveats. But in the future, what I see is, and again, this is just, there's no concrete prediction here,
yeah,
but what I envision is the early phase of drug development getting faster as AI is applied to the molecule design and
yeah,
preclinical aspects, and to extracting more information in the clinical development process from each subject to be used in decision making, as I described. And then in the later phases, AI being used to make trials more efficient in terms of both the workload, the resourcing, as well as insights about things like recruitment. Where are you likely to find the right kinds of patients to enroll, and things of that nature. So what I see is an acceleration of drug development through each of those stages that AI can enable, as long as it's done, with the appropriate ethical safeguards and
yeah.
the appropriate scientific rigor.
Absolutely, and it'll help bring drugs to market faster if we
Right.
are able to envision that. Yeah. Thank you, Greg. Really appreciate it. These were all very, very insightful thoughts that you have provided in this podcast. Anything like you would like to say in closing?
No, thank you very much for inviting me, and I look forward to seeing some other guests on your show now
Absolutely.
that I'm aware of it.
Yeah. Thank you, Greg. Really appreciate it.